All right. Chapter 11 is a lot, you guys. Most textbooks break chapter 11 into several different chapters. Like there's one for replication, there's one for protein synthesis, there's one for mutation, but the OpenStax textbook puts it all into one chapter. So this is a lot of information. The beauty of this is you should have all had some of this in previous coursework. You should know the basics behind DNA replication, and you should know what transcription and translation means. You should know a little bit about DNA mutation. This chapter should we're going to take it a little bit deeper than you've had in previous coursework. Since you should have had some of this stuff before and we're just going a little bit deeper, I'm really going to not talk a lot in this little video, and I'm going to say go watch pre-existing videos because there's a bunch of good stuff out there behind all of these topics. So the first thing this chapter does talk about really is DNA replication. And like I said, there's a bazillion decent videos out there that show you how DNA replicates. The last chapter we talked about the chemistry behind DNA, so make sure you remember that stuff. It's a nucleic acid, it's made out of nucleotides, each strand is a polynucleotide covalently bonded together. The two polynucleotides are hydrogen bonded together. The A's and T's get 2 hydrogen bonds between them. The G's and the C's get 3 hydrogen bonds between them. That's relevant for the origin of replication. The 3 steps for DNA replication are initiation, and that happens in the case of E. coli, at OREC, which is the origin of replication. They have that one origin. Now, eukaryotic chromosomes, they tend to have lots of origins spread out along the linear chromosomes, and so that is one of the things that is different between prokaryotes and eukaryotes. Once we have initiated at wherever the origin happens to be, we elongate from that point. And the origin, since it's in the middle of something somewhere, we're going to elongate in both directions. So if you're looking at this strand, we're going to go from both the top and the bottom, wherever we originate at. And then we're going to terminate once we hit the end of a linear strand, or once we've come back around to the beginning of a circular strand. Now, because I didn't like the picture the book gave you, I gave you this picture from a different textbook. It shows you the replication fork. This is created by the enzyme helicase. It separates our 2 DNA strands, and then we get this whole replication machinery that comes into place. We get DNA polymerase that comes in, and it's going to start adding nucleotides one at a time. It's going to build one strand continuously, and so the strand that's built continuously, we're just going to keep adding nucleotides. The replication fork is going to keep being stripped in this direction, and this strand is not going to have any breaks or cuts. It's just one nice continuous strand. The other strand, on the other hand, gets built in these little pieces, and it's being built in the opposite direction because of the bidirectionality of DNA. So instead of us adding nucleotides up here, DNA polymerase can't do that because this is the wrong side. It can't add to the wrong side. And so it can only add to this side of the DNA molecule, which happens to be the 3' end. So once it hits the end or a pre-existing DNA fragment, it stops. We restart from the primer that primase added, and then we build another fragment, then we'll stop. We'll start again from another primer. We'll keep going until we hit the next strand and we stop. So we have all these little fragments that are called Okazaki fragments. Those fragments have to be glued together. We have to remove all the primers. It's a whole thing that has to happen. And so make sure you understand what I'm talking about when I say primers. Make sure you understand the Okazaki fragments. Make sure you understand all the enzymes that are coming into play, from helicase to DNA polymerase 3, DNA polymerase 1, ligase. I think I said primase already, but just in case I didn't, make sure you know what primase is doing. I linked a video in the PowerPoint. I can't show you this video here because then it would get blocked on YouTube, but do make sure you watch that video. I've also linked a couple of other videos that can help you understand in pretty good detail what's going on behind DNA replication. Okay, after replication, it gets into the central dogma of molecular biology, which is essentially just protein synthesis. There are 2 steps to protein synthesis: transcription and translation. Transcription is essentially like DNA replication, only instead of going from DNA to DNA, we go from DNA to RNA. Also, instead of copying both strands of DNA, we're only copying one strand, and we're not copying the whole strand. We're only copying the segment that we need to which is the gene that we need to copy at this one time. So there's similarities and there's differences between DNA replication and there's transcription. After we have transcribed a gene, the transcript, which is usually messenger RNA, is going to go find a ribosome. The ribosome is going to read the messenger RNA and then make a protein product. Now, proteins start out as polypeptides. The polypeptide then gets processed. Processing involves folding it into the correct secondary, tertiary, sometimes quaternary structure, and so all of that is protein processing that happens after translation, which is what the ribosome does to actually make the protein. Again, you can find a lot of good pictures and a lot of good videos to show you how this is going to happen. Make sure you are able to transcribe a strand of DNA if I tell you to. Make sure you are able to translate a strand of mRNA if I tell you to. Let's see. Stages of transcription basically follow the same steps that you had for DNA replication, still initiation, elongation, and termination. But instead of initiation happening at an origin, it happens at a promoter. Elongation does not happen until you get to the end of the whole chromosome. It happens when you get to the terminator, which is at the end of the gene. And so we elongate until we get to the terminator at the end of that gene. So again, there's similarities and there's differences. And again, I have linked to a video. You can click on this video in the PowerPoint that's available for you guys in the learning management system. I just can't show it to you here because again, it would get blocked in YouTube. After the protein synthesis part, which again, you need to really make sure you understand it really well, because if you don't understand that, you're never going to understand why mutations matter, and you're never going to understand operons. But mutations are changes in the DNA sequence. Mutations happen all the time because DNA polymerase is not perfect. So when DNA is replicated, it makes mistakes. A lot of times the mistakes that get made don't matter. Those are called silent mutations when that happen. Which is the top picture that you have there. So in this case, it changes this A to a T. That does not change the amino acid sequence, which means the protein does not change. And so a lot of your mutations are silent mutations. A missense mutation happens when a letter changes and it ends up changing one of the amino acids in a protein. This is what happens to create like sickle cell anemia. We changed one amino acid, and as a result, the entire hemoglobin molecule is broken, and now the person has a disease as a result. So sometimes a point mutation or a base substitution can result in a change in the protein. Sometimes that can cause disease. Sometimes that can be advantageous. It just depends on what we change this ingredient to and then how that alters the actual protein. Now, a nonsense mutation is almost always very bad because let's go back to hemoglobin. It is a very long protein. Let's say that in the fourth amino acid, where we should have been continuing transcription, the fourth amino acid changes to a stop. So instead of having hundreds of amino acids, it's now 3 amino acids long. That means this protein, instead of being functional, it's now a nonsense sequence of 3 amino acids that are more than likely never going to have any function inside of the cell. That's why it's called nonsense. It changed what used to be a nice functional hundreds of amino acid long protein into just 3 amino acids that have absolutely no function inside of the cell. So it greatly truncated the protein, essentially. Oh darn, I did the wrong thing. Sorry, I was going to scroll down in the picture and I can't do that. You can hopefully see the whole thing anyway. Frame shift mutation. So everything in DNA gets read 3 letters at a time. Everything in RNA gets read 3 letters at a time as well. So in this DNA, what happened was a deletion. Actually, 2 letters were deleted. The T and the A went bye-bye. And so as a result, everything else got shifted 2 letters over. And so what that meant was, as this got transcribed into mRNA, what used to be this UNA became GCC, so that changed this valine to an alanine. That also means all of the other amino acids past that deletion are going to get changed. And so everything past that point changes. This is usually also very, very bad because it changes every ingredient in that protein. Every now and then, though, something wondrous happens and we make this new protein that is magical, but it's almost always very, very bad when we do a frame shift mutation for a protein. Okay, After that, your very brief, how do we control gene expression? In prokaryotic cells, they have these structures that are called operons. So an operon is the promoter, which is where RNA polymerase binds so that it can start transcription. An operator, which is where a transcription factor can attach so that we can regulate gene expression. And then all the genes that get regulated by this operon, this operator, essentially. And so this is what we're talking about here when we talk about an operon. It's all of these parts plus the genes that get regulated by that. The two that I wanted to go into a little bit of detail with you guys on are the trip operon and the lac operon. So the trip operon is called that because it contains genes that regulate the prokaryote's ability to make the amino acid tryptophan, which is one of the 20 amino acids that are necessary for making proteins. So here's how this is going to work essentially. The normal state for this operon is it is normally turned on, which means RNA polymerase normally binds, we zip through here, we make all of these gene products, the cell can make tryptophan, life is nice and happy. This is a repressible operon because if the cell already has plenty of tryptophan, why would it need to make more? So tryptophan will come and it will bind to the repressor. That will allow the repressor to rebind to the operator. And as a result, it basically puts this gate up so that RNA polymerase can bind to the promoter, but it can't zip through here because there's a gate up. So it can't actually transcribe any of these genes. So we won't make any more tryptophan because there's already plenty of tryptophan in the cell. So this was your example of a repressible operon. Next up, we had your example of an inducible operon, the lac operon. It's the exact opposite. So this one exists in a state where the repressor is normally pre-existing attached to the operator. So RNA polymerase cannot normally make these enzymes that these genes code for. Because lactose is not usually available to this organism, why would it need to make the enzymes to digest lactose? If there is lactose available though, lactose binds to the repressor, changes the shape of it so that it detaches from the operator. Now RNA polymerase can bind to the promoter and zip down and go ahead and make these enzymes so that we can digest lactose and go ahead and make energy out of lactose at that point. And so that's the example of an inducible operon that you guys have. Incidentally, scientists were really excited when we figured out how prokaryotes regulated their genes. Operons were so elegant and they were so simple. And then it turned out eukaryotes do not regulate their genes like that at all. Eukaryotes like us, we don't have operons. Instead, we are way more complicated and we are still trying to figure out how we regulate our genes. We know that it's not actually the DNA itself, like an operator is part of the DNA itself, that regulates our genes. Instead, it's something that we put on top of the genes, which is why we call it epigenetic regulation when it comes to eukaryotes. What happens is we can add methyl groups to our DNA or to the histones that we pack our DNA around. And generally speaking, the more methyl groups we add to the DNA, the more repressed we have made the DNA. However, that is not always the case. As always, eukaryotes are just more complicated, and this is a gross oversimplification. But generally speaking, if the DNA is methylated, we have turned off the genes that have been methylated. Hopefully that gives you a decent summary of what's going on in that chapter. Again, I know it's a huge chapter and there is a lot going on in it, but hopefully that's a decent refresher to get you your basic information that you had from previous semesters. Good luck getting into those bigger details though.